Okay, so let me try to pull up the whiteboard. Where can I do it? Annotate, tabs. Remote control, share. Okay, I see. Probably here. Maybe it's the sharing under. Okay. Like this. Okay, so if I'm going to use the whiteboard to write something here. So the first question we would like to discuss is, make it larger. Is this inference versus prediction? Okay, so what is inference? What is prediction? I think the narrow sense of statistics and machine learning, a way to differentiate the two is what you just mentioned, right? Typically, when people say inference, it's about using your data to infer something about the population, or using Weijian's language, the population is what we thought as the knowledge, right? What is the underlying biological truth? I think for most scientific research, the problems we handle are of this type. The reason is that we don't think our data is perfect. We know the data is always a mix of signal and noise, right? Then how can we disentangle the signal and noise from say something about the knowledge? And I trial. So whenever we do clinical trial, we will never get to test our drug on everyone. We only have a random sample of representatives, right, to participate in the trial. And we hope that the effect of the drug we measure on this sample, these patients, would be informative for how the drug will work on the population. So that's the inference. So statistical inference, as you can see from this point of view, is both about a point estimate, which is what is the effect of a drug, my estimate, and some uncertainty quantification. How accurate do I think my estimate is? So it's always an estimate and some error bound. So that's why we often provide a confidence interval for the output. So it could be in the example as a confidence interval. So this is what people use in the frequentist statistics. So what it exactly means is that in the frequentist sense, I'll just quite quickly try to go over the overview of many things. I know it's maybe a lot, but I'll try to be as simple as possible. So in the frequentist sense, we believe that data is random, population is fixed. So that's the most natural way of thinking about sampling. Then the confidence interval is considered to be an interval we construct from our data. So for every data set, for every sample I got from the population, I will get one confidence interval. And my hope is that if I get many samples and many confidence intervals, then these confidence intervals will jointly cover the truth, the knowledge with good probability. So basically, when we say confidence interval, when we say 95% confidence interval, that probability is not about this one particular interval you get from data, but it's about this collection of all possible intervals using the way you do the interval construction. So it's about a confidence interval construction. It's not about a particular interval. And that's the most confusing part of it, because many people will think, oh, given this interval, then my parameter, my truth, will have 95% probability falling into this interval. That is the most common misconception. The reason is we are not fixing the interval. The interval is just one of the many possibilities. Okay, that's the frequentist sense. But we also have and I'll just say, aligned with confidence interval is the concept of p-value. Another thing many people would tend to misunderstand. So way to think about p-value is just to think about it as a transformation of your data. It's a data summary. We summarize your data, your sample into a p-value. Of course, the p-value is constructed based on your null hypothesis. But the null hypothesis is a question you set up before you see your data. So the null hypothesis is not from your data, but it's a question you want to answer. For example, if the drug has any effect, right? So the null hypothesis would then be the drug has no effect, right? This is a question we set up or hypothesis we want to reject before we have any data. Now with the data, we summarize our clinical trial data, the effects we measure into a p-value, in the hope that the p-value will give us some indication about how likely we're going to reject the null hypothesis. So the intuition is that the smaller the p-value, the more likely we're going to reject the null hypothesis. So the tendency, we want to reject the null hypothesis. So consider p-value just as the data evidence, the evidence you extract from the data against your null hypothesis. So again, in the frequentist paradigm, p-value is random because your sample is random. Okay, that's the frequentist. On the other hand, we also have similar things for Bayesian. For Bayesian, we use the terms like credible interval, and we have this thing called Bayes factor. So you can see that I'm not giving you any formula for today, just to give you the intuition behind these terms. So for Bayesian, the thinking is the orthogonal. So basically, what we see as Bayesian is, okay, the data is our evidence. Conditional on data, what can I say about my truth? So Bayesian thinking is that the truth can have many possibilities. It's not one single fixed truth. But we don't know what possibility the truth is. Therefore, we want to use our data to tell us which possibility is more likely. So for example, the drug effect may have a distribution from 0 to some maximum, but it has a distribution. But I want to know that given my sample, given these people I recruited, what is my guess of the drug effect? So in this case, in the Bayesian inference, everything is about this so-called posterior probability, which is the posterior means that, okay, the distribution of my truth given my data. So that is what we care about. And based on that posterior distribution, we can have credible interval. And the credible interval is very different from the confidence interval because in the sense of credible interval, we fix the interval because the interval is based on data and data is considered to be fixed in Bayesian paradigm. The interval is fixed, but the parameter, the truth is random. Then the parameter or truth will have 95% probability to be in the credible interval. So you can see that it's orthogonal thinking, which is random. So if I want to draw this, is that one dimension is data, the other dimension, okay, so I'll just type. So one dimension is our data, and the other dimension is our parameter. So what is the difference is that, what is different is, let's see, oops, I don't know why I have this error here. Okay. Oops. Okay, so let's just stick with this. So what we are trying to say is that when we do frequentist inference, we are fixing the parameter. We are fixing the parameter and do something, talk about the data. So that is the frequentist. But when we do Bayesian, we are fixing the data and talk about the parameter. So it's this way. So therefore, I think I'm getting into something quite, let me see, quite challenging of statistics. I'll try to move here. which has been a century-long debate between the two paradigms. But for application, people may not really, you may use a method without thinking too much about this fundamental difference. But in fact, I think this is very important to know, since it's about how we infer the truth from the data. It's a different orthogonal perspective. So therefore, I would say the difference between frequentist and Bayesian is philosophical. And I would say for most people who focus on algorithm or algorithmic aspects, or I would say in computer science, most of the focus is algorithmic. People are more aligned with the Bayesian perspective because you take your data as given. You apply operations to your data, right? And the algorithm will run on your data, give you some output. So I think the Bayesian paradigm is like, and also many biologists will tend to think like in the Bayesian way because they trust their data. They want to use the data as the condition, right, to make some inference. But frequentist is more about, okay, I want to say the data is just one of the many possibilities. So therefore, which one is more reasonable? I think. But if you are someone who studies statistics, people may have very strong beliefs to be for one or the other. And as I said, in the past century, past half a decade, not half a decade, 5 decades, 50 years, there have been many interesting papers discussing the pros and cons of the two paradigms in different scenarios. So the short message is that there's no perfect solution. Because for Bayesian, you always need to assume some prior distribution. That is the thing many frequentist people criticize about. How would you trust your prior? Is it subjective? Is it just for convenience? And for frequentists, there are some cases people think are kind of counterintuitive. For example, as you will see, that the p-value would be something that depends on your sample size. So we know that when our sample size is large, you can easily get a very small p-value, even though the null hypothesis doesn't seem to be that unreasonable. So you will have a stronger evidence reject the null. And the reason for that is because in the testing framework, null hypothesis versus alternative hypothesis, it's asymmetrical. By asymmetrical, I mean that you're not treating the null and the alternative to be equal to each other. You have a preference. Your preference is that I stay with the null hypothesis and only reject it unless I have very strong evidence, right? So that asymmetric nature will give you something you may find unnatural. So that's some criticism. But just stepping aside from the debate, because I don't think there's any possibility to reconcile since it's orthogonal. But I think from an application side, I will say many methods you see in bioinformatics are Bayesian. The reason is just because Bayesian makes the statistical inference problem more of a computation problem. So for example, in R, you now have a message language nested in R called Stan. Stan is the R language specific for Bayesian computation. So anything like MCMC, which is just trying to do sampling from the posterior, becomes automatic. And now I think in Python, there are also some package for doing the variational inference, which is another jargon, but basically it's based on Bayesian. So variational inference is about the fact that, okay, when the posterior is difficult to sample from and MCMC is slow, let's just sample from some proxy of the posterior, an approximation of the posterior, not the original posterior. Then you get faster computation. So you can see that it changes the problem to be computation. Then it makes a lot of the problem-specific method development more automatic. But I would say that maybe because of training, since I graduated from Berkeley, and Berkeley is more on the frequentist side, I would say that for frequentist solutions, in many cases, especially the classical ones, right? T-statistic and Wilcoxon's test. So these are all derived under the frequentist paradigm. I would say if you can have a frequentist solution, it's usually simpler because you may have some closed form to play with, but yeah, but it's more specific. So you have to develop something specific for each problem. So I mean, I would say the developmental process is not as automatic as Bayesian, but if it's possible, you may get a simpler solution. Anyway, so I think that's a very lengthy introduction for this part, inference. Then I said inference versus prediction. What is prediction then? So for prediction, it's mostly about one data point instead of the population. So in this case, we would say it's one data point, and there are some terms referring to the same thing. I think in computer science, machine learning, people say one instance, and in statistics, people say one observation. But they're all about the same thing. It's just one draw from the population, and you want to make a prediction of it. So specifically, when people say supervised learning, it's all about this. So still using the drug as an example, then for prediction, we are interested in, okay, if I apply this drug to you as a person, what will be the effect? It's not about the whole population, but about this one person. Okay, so therefore, you can see that the objective is essentially kind of different, because for prediction, as our goal is just to predict well, this becomes more like a task in which we can somehow tolerate the prediction algorithm being a black box, right? As long as the algorithm gives us very nice prediction results on a large enough test data set, we think it's working, right? That's something deep learning has been super successful, and people like that for image classification, right? And now for the basically, I think image classification, and also for annotating the language, right? What is the tone of the text? Is it negative or is it positive? For those tasks, is prediction tasks. But for inference, right? If we are thinking about what we really want to infer as the knowledge, then the model or the method itself should be something interpretable. So we need to interpret what is really going on there. So I think in the ideal sense, when I try to interpret a model, we want the model to have good prediction accuracy, so we can say, okay, the model is a reasonable model to interpret. But in many cases, especially as now the task or the data sets getting more and more complex, I would say many methods are on one side versus the other. Like it's more about the inference or more about the prediction. But ultimately, I think trying to unify things would be our goal. Okay, that's the first question I heard from the keywords. I think the second keyword I heard is sampling. I think several of you said that. Can you raise your hands if you mentioned sampling? Okay, only one. I thought there was another, but yeah. So for sampling, that's a very good term. And let me try to put it here. Sampling. Why do we need to do sampling? So Weijian, what do you think? What's your interpretation of sampling? We don't want to go through all. Okay. When we sample them at representation. Like speed up everything. Okay, so you're talking about the practical aspect of sampling. So it's for okay, let me see. So what you talk about is for kind of numerical approximation, right? For computational efficiency. So this is actually subsampling, right? You sample from a large sample. Okay, so yeah. And another way I said for numerical approximation, maybe I should put it down here, is what we have for MCMC. Okay, so for MCMC and variational inference, the goal is trying to avoid the closed form computation evolving posterior. We try to use the sample to represent the distribution and do some computation. Okay, so this is basically what people call Monte Carlo computation. So for those of you who have never heard of Monte Carlo, what it means is that if I want to compute some average of the distribution, but I don't have the tool to do the integral, because if you have the distribution, say as a Gaussian distribution, then to get the exact mean of it, you need to do the integration. If you don't have the tool, what you can do as an approximation is to just draw a sample from the Gaussian distribution, set a sample size to be large enough, say 1,000, then compute the average of the 1,000 values. That is an approximation to the expectation of Gaussian. So that's Monte Carlo approximation. Okay, and so basically this is for practical use, right? But I will say another thing when people need to do sampling is a very important aspect of statistics, but it has been largely unmentioned nowadays, is experimental design. So experimental design actually is the first step in our data collection. We need to think about how to design our experiments so we can collect data, so the data will be of good quality. So the short message I can share with you is that if your data collection is perfect, then you just need simple statistics. Yeah, a lot of the things we develop for handling those difficult situations because the data collection is not under perfect design. But I would say that perfect experimental design is a luxury. It only happens when you are doing wet lab, right? Experimental science, where you can afford to do, say, cross design, so you can actually use the design itself to cancel out many confounders. But in many cases, the data we get are observatory, or people say observational. By observational, we mean that there's no design. We just collect whatever we have. And this is especially the case for genetic study, right? For those, say, UK biobank studies, you have to measure whomever participate in your study. So therefore, the data may have many confounders. So that will make statistical thinking more important. So yeah, so that's what I want to say. So, but once you have the design, then you sample from, your measurement will be just a sample, right? Okay, so that's the second keyword. And I think I've also heard supervised learning, unsupervised learning when we discussed. So let's move to this side. Let me see check the power. I don't think that can do this. Seems to be. So for these two, when we talk about learning, and the term learning is from machine learning. So it's the same learning term. And what it's really about is the focus is still on this one data point, one instance, one observation. So what it actually means is that we care about the data point xi. I'll just put it as, for supervised learning, we care about this xi, yi. And for unsupervised learning, we just have one instance xi. And please note that here I'm using this uppercase for x because typically the xi is referred to as a feature vector, and it has multiple features, so it's a vector. It has multi dimensions. It's not a scalar. Feature vector. And there's another interesting difference in the tradition. That is, in statistics, people like to say it's p-dimensional. But in machine learning, many people use d-dimensional. So you will actually see this difference in the notation convention. But yi is often a scalar. Okay, so if it's categorical, people call it a classification task. Or maybe I could say discrete, because it could also be ordinal. Ordinal means the classes follow a certain order. And or continuous. If it's continuous, then it's a regression. Okay, so the availability of yi is the difference between supervised learning and unsupervised learning. For unsupervised learning, we only have the feature vector for each instance, and then we want to talk about it, right? So for the supervised learning, the goal is to predict yi from xi. Okay, so just say that in this case, we want to find some function. So the key thing is that you want to find some function fxi, find f function such that fxi is close to yi, right? And that's the goal of supervised learning. But to define the closeness, what do we mean by close? There's another thing to consider. That is the distance metric. How do we measure the distance between fxi and yi? And typically people use the squared difference. Okay. So that's the supervised learning part. And for the unsupervised learning, I think some of you already mentioned, there are two major tasks. One is called dimension reduction. And typically the goal is to project your xi from a p-dimensional space or d-dimensional space, which is a high dimensional space, to something zi in the two-dimensional space, R2. That's dimension reduction. Or typically, I wouldn't say R2, maybe RK with K much less than P. So like PCA is a typical way to do dimension reduction. And the goal there is we believe that the data is not so high dimensional. So the key information, the signal can be summarized in a much lower dimension space, which is k-dimensional space. We think it's kind of a denoising step, right? So for example, PCA is kind of a denoising step for one use of it. It's a very powerful method for many users. Or if we have k equals 2, then it's typically for 2D visualization. And in the single cell field, people typically use two methods, t-SNE and UMAP, for this purpose. And this uses just to look at our data. Otherwise, we cannot visualize a p-dimensional space, but we can visualize a two-dimensional space. That's the first unsupervised learning. And the second unsupervised learning is clustering. So we don't have yi, but we hope we could potentially, we could group our instances into groups so that the instances in each cluster share similar feature vectors, share similar x's. Okay, so that's another aspect we want to discuss. So basically, for supervised and unsupervised learning, as you can see, we haven't really talked about or focused on any parameters or truth, right? We're not saying that. So supervised, you're still concerning each instance. Unsupervised, you're also concerning each instance. But of course, you can always use your statistical thinking or model perspective to develop something based on a model. And that's what people call model-based clustering. Right? By model-based clustering means that you believe there's some underlying true model in which the instances are coming from a model with certain components, and these components are the true clusters you want to recover. If you have a model for that, then you can have some way to evaluate your clustering algorithm. Okay, but the clustering algorithm itself is just an operation on your data, on your sample. It's not about the population. Okay, so that's the third aspect I want to talk about. So as you can see, probably the take-home message I want to share with you today is that we need to have this clear distinction between instance and population. So whether we are talking about specific data points or we're concerned about certain aspects of a population. That's the thing I want everyone to think about. And also model versus algorithm. Maybe I'll just go down and put it here. Yeah, model is itself another jargon which may mean different things in different fields. What is your understanding of a model? Maybe I can have one volunteer to give me one answer. What do you think as a model before you take this class? Maybe one question for you. Do you think random forest is a model? You think it is? Okay. How about others? Yeah. Okay, so you would say that a model is a relation of variables. Okay, so how the variables are related to each other, you call that a model. Okay, and yeah, so yeah, back to that. I think I know some people might say random forest is a model, but I think in the narrow sense, I would not say it because it's an algorithm, and using a random forest, you don't actually have a pre-specification of the model for, right? You don't say, okay, what are the things because for each random forest, it consists of each, right? And each I can possibly call it a model, but I think for random forest, anyway, so that's something debatable. But I would say in the very, very narrow sense, maybe the narrow sense in the statistical language, when people see a model, it is a probabilistic distribution. So it's a probabilistic distribution you can sample from. You can draw a sample from. So I would say narrow sense model is a probabilistic distribution. Yeah. And for distance, actually, the large language model now people talk about is a model. The reason is that we can sample from it. So every sentence, for example, generated by ChatGPT is actually a sample from this super complicated model. It has maybe 50 billion parameters, 100 billion parameters, but you can sample from it, right? So, and actually, the training of large language model is unsupervised. We didn't have labels of the sentences we input into the training. It's just trying to learn the relationships between words in a very long range. In a simple language model, you just learn what is the next word. You calculate the conditional probability of each word for its next word, right? So then that will be the transition probability, just like the Markov chain. But for the large language model, you go much longer range. It's not just the nearby words. Maybe you can be 1,000 words apart, but you try to learn these complicated relationships. And the goal for training is that we want each input sentence to have a large probability to be output based on this model. So it's still the maximum likelihood principle used for training, and which we'll talk later. But basically, it's still a probabilistic distribution for which you can sample from. And whenever you have a probabilistic distribution, the key thing is to specify your variables. You need to know what variables are there. And then given the variable, for example, height is a variable, and let me say age is another variable, right? Then you can say that we can use these variables to specify our whole human population. Then every person can be considered as a sample, a draw from the human population. That's just the sampling perspective. But for algorithm, it doesn't have to be, you don't have to specify what is the variable. It's basically operation. It's just an operation on data. So for example, I give you one matrix, and if you want to do modeling, the key thing is try to understand in your matrix, what are the instances, what are the variables. So for example, in the gene expression matrix, for example, I have, say, RNA sequencing data from many biological samples measured for these genes, right? Then in this case, what would you consider as variables versus instances? That's another key distinction we need to make first before we do any data modeling. The question is, if I give you a sample by gene matrix from RNA-seq data, would you say whether the rows as samples, biological samples, if they say I have samples from 100 individuals, RNA-seq data, and the columns are the genes, then which direction whether the samples or the genes should I call variables? Yeah. Genes should be variables. And why is that? Yeah. So basically the variable is the thing that's unchanged, I would say. So basically genes are genes, right? You get the same genes in everybody, but expression level changes. So when we say variable, we need to think about it as a quantity that can be measured in different samples, that can be measured again and again. But for samples, we need to connect it to population. Do you think your samples are just a random set of observations from a whole population, which is the case for your biological samples, because we can do our experiment again and again to collect more samples, right? But the genes we measure are not a random set. It's not like if we do more and more experiments, we can get more and more genes. It's not like that. So that's why I think considering that difference between genes and samples, or genes and observations, or variables and observations, is the key thing. But if you just develop an algorithm, you may not need this distinction. What I mean is that you may cluster your rows, you may cluster your columns, right? In that case, you're treating your rows and columns as just symmetrical, or you don't consider them differently. So therefore, you can do clustering. From a mathematical point of view, it's a matrix. You can do transpose, right? But from a statistical point of view, it is very important to distinguish the two. So therefore, I think for week 1, I will post some of the papers for your reading. For this week, we'll just do some reading, because I think these are interesting papers to read. And one paper I wrote 3 years ago is this thing. Try to offer some perspectives on the distinction between the hypothesis testing in statistics versus the binary classification in machine learning. So the second is a supervised learning task. The first is an inference task, because both are trying to make some binary decisions, right? Whether I reject a null hypothesis or not. For binary classification, we try to assign each instance a label. So, but they are fundamentally different. So I try to summarize these aspects in this paper, and I'll leave that for your reading, because I think this is mostly a graduate class, right? Even though we have 2 undergraduate students, but as a graduate level class, I think we will rely a lot on your reading and self-learning after class to gain the most out of the class. So just to maybe, since we are already here, I'll quickly say that the guidelines to differentiate the two, like the first guideline I offer is trying to decide on, oh, I think I should change my screen sharing because they cannot see it. But how can I share this? Share whiteboard. Do you know how I can save it? Yeah. Export. Okay. pages. Okay. Okay. So now I will change my screen sharing. Close to this. So I was talking about this paper for people on Zoom. So the first guideline is that we need to decide on instances and features. And the second guideline, maybe I can go back to this figure here. Yeah, so this is the table matrix we are often given to analyze. So this is an example in which we have patients and genes, right? Which is the same as the example I gave. So we need to first decide we treat patients as instances and the genes as features. And using this, I try to give an example about the difference between hypothesis tests and binary classification. So for hypothesis tests, we're asking questions about the population. For example, here is if my patients have disease patients with the disease, yes. Without the disease, no. And some patients without a label marked as unknown. Then for hypothesis testing, we're concerning questions about genes because every gene as a variable, it has certain properties in the population. Then, for example, this question could be, if is gene A a biomarker? The question is, can I use gene A to distinguish the two groups of patients, disease versus undisease? Then this is an inferential question, right? And this null hypothesis is that the gene is not a biomarker. Then I will pull out patient data for this gene, this vector, then I do a statistical test to compare yes group versus no group to reach a decision. That's the inferential question. The question is about genes. But for binary classification, it's about instances. So therefore, if my question is to answer whether this unknown patient is disease or undiseased, then I will use these patient with labels to train a binary classifier, then apply it to this left out unknown patient to do a prediction. So I think this is something I want to emphasize in this perspective article, because in bioinformatics, sometimes we do see people confuse the two, honestly, because I've seen cases where it should be a binary classification question, but people are trying to calculate the p-value. Right? They're trying to calculate one p-value for each instance. And that's actually the motivation for this article. Since we are already here, maybe I can say a little more about this article. It's about this cancer driver gene prediction. Okay, so this is what I was motivated to write this for. So in the problem cancer driver gene prediction, the goal is to utilize an individual gene's mutational signatures, such as the number of disease missense mutations, to predict how likely the gene drives cancer. So it's a cancer driver gene prediction problem. Note that the mutational signatures are aggregated from multiple patient databases, and individual patients' data are unavailable. We have knowledge of a small set of cancer driver genes and neutral genes that are unlikely to drive cancer. The question is, how can we leverage this knowledge to predict whether a less studied gene is a cancer driver gene? A famous algorithm, this algorithm Tucson, addresses this question using a hypothesis testing approach. In brief, it regards mutational signatures as features and uses hypothesis testing to assess how much an individual gene resembles known neutral genes based on each feature. The gene's feature value is used as the test statistic whose distribution under the null hypothesis is estimated from feature values from non-neutral genes. From the test statistic and the approximate null distribution, the gene receives the p-value for that feature. Suppose that there are 10 features in total, then each gene receives 10 p-values, which are subsequently combined into a single p-value by features method. Actually, I have some talk slides to show this better. So basically, I can quickly go to my talk. And this one. Yeah, So this is what I'll try to say. So basically, what it happens in this paper, it was a cell paper. What it does is that and they have genes here, features here. So the features are the genes' mutational signatures. They want to predict cancer driver gene. So what they try to do is that they take each feature, say feature 4, they compare this gene A, they want to predict whether it's a cancer driver or not, take its value to compare it with the null values, the same feature for non-cancer driver genes. Then you try to get a tail probability. So as the p-value. So for example, if the cancer driver genes, the blue ones for feature 4 are on the larger end, larger than the null values, then they will calculate the proportion of null values greater or equal than gene A's value as the p-value. And if the cancer driver genes values are less than non-cancer driver genes, they will calculate the proportion of null values less or equal than gene A value as a p-value. So they'll get one p-value for gene A for feature 4. And they do that for all features. If we have a D features, then gene A will get D p-values. Then they aggregate these p-values into one p-value using Fisher's method. And they do that for each gene, PA to PZ. Then they try to find the p-value cutoff to call cancer driver genes. So that's what's in the Cell paper. But I think it should be a binary classification problem. So in other words, I think what we should do is that to try to use all the features jointly to train a binary classifier based on the cancer driver genes as one class, non-cancer driver genes as another class, train the classifier, apply that to each unknown gene to make a prediction. And that's how we get this collaboration, this work, this method. So we implemented this binary classification. But the original motivation is not to change the methodology, but try to use epigenetic features on top of genetic features. But when my collaborator Wei Li talked to me, I had this question about this approach, so we changed it. So then back to the paper I was talking about, why this is problematic, right? Why this approach is problematic. So let's see, from a statistical perspective, there are 4 apparent issues with this hypothesis testing approach. First, each hypothesis test, one per gene per feature, only utilizes the non-neutral genes to estimate the null distribution, but does not fully capture the valuable information in non-cancer driver genes. To be exact, non-cancer driver genes are used to select the predictive features before hypothesis testing is performed. However, in each test for one gene and one feature, the information of non-cancer driver genes is only partially reflected in the direction of the gene's p-value, two-sided, larger than, or smaller than, which excludes the possibility that the non-neutral genes may have feature values on the two sides of the non-cancer driver genes. Second, each hypothesis test is performed using a sample of size 1, because here the test statistic is the feature value of one gene, which is known to be not powerful and thus undesirable. If the gene is a cancer driver gene, we may miss it with a high chance. This is the reason why we recommend using more than one instance for hypothesis testing. That was in the guideline. Third, combining multiple p-values into a single p-value is a difficult task, especially when p-values are not independent of each other. The fact that mutational signatures are observed to be correlated features, their resulting p-values are correlated for each gene, violating the independence assumption of Fisher's method. Although there are methods for combining dependent p-values, they cannot address the most fundamental question: what is population behind each hypothesis test, leading to the last issue. Fourth, the population behind the null hypothesis is unspecified. For a given test, is the population about that particular gene or all the genes? Therefore, we conclude that this hypothesis testing approach is inappropriate for this cancer driver gene problem, which instead should be formulated as a binary classification test by the reasoning below. Yeah, so basically, I think this is a very typical example I want to share with you. Maybe I think in other communities, people might be confused about why the Cell paper would go with the hypothesis testing approach, right? Why would you compute one p-value for each gene? But in the bioinformatics approach, this may not be so surprising. I think the reason is that people are so used to p-values. people might think, okay, I need to get a p-value anyway, right? Regardless of whether this is really an inference problem or hypothesis testing problem. So therefore, I think my take-home message for you for the first lecture is that we should be thinking about the difference between inference and prediction. That's the most important thing. Secondly, when we are given a data set, what are the variables? What are the instances? Yeah. So I think that's for the plan for today. And then coming back to the syllabus before we wrap up, I forgot to talk about the syllabus yet. So for the syllabus, for this quarter, as some of you mentioned, since I have recorded lectures before, I wonder maybe I should do something new instead of repeating myself. So I think I mentioned to some of you a while ago, I think Bayesian may know this, is that I plan to write a book, but I was so delayed in the writing process due to other duties. But for the book, what I hope to do is to use the popular single cell data analysis pipeline Surat. I think most people use it for single cell data analysis, and it has many, many steps. So I want to use Surat as the context to explain statistics. And that's what I hope to do this quarter. Like, why does Surat would do highly variable gene selection? Why PCA, right? For every step, I hope to provide some reasoning behind it. So that's my new plan for this class. And also, I hope to get your permission for a project, for the class project. So it's just like homework and project. And I think for the homework, we will try to ask you to do something based on the class lectures, do some tries to see the effects of the steps. But for the class project, I'm interested in trying an experiment, social experiment. So the hope is that we will provide you data set for you to analyze, but don't worry about whether your analysis is correct or wrong, because we want to give everyone some fair chance to do the thing you really believe in. So basically, everyone will receive the same grade. So don't worry about the grades for this class project. But I think I want to see how people's analysis may differ on the data set. So I think this is a way for us to reflect like the diversity in single cell data analysis result. Because I do have this question that given so many steps and so many options, the results could be very different, right? If you are not given the hypothesis beforehand, okay, what I'm looking for, then try your approach, try to see what you can discover from the data set. I think we will see something very interesting, like people may get different things. But try to make sure that you don't worry about the grade, so we will give everybody the same grade. So that is a chance for us to really explore the diversity of data analysis. So that's the current plan. And I'll try to give you the data set maybe as early as possible. So you should have at least before week 4 for sure. So you will have enough chance to play with the data for the final project. That's the plan. Okay, so then I'll see you on Wednesday.